So when it comes to the plasma membrane, we also need to take into consideration the fluidity of the plasma membrane, which is very important. It's basically how resistant to the temperature in the surrounding and the higher temperature is the plasma membrane. So you're going to see that there are different in melting and boiling point when it comes to the composition of the phospholipid bilayer. And you're going to see that, for example, fish in the ocean that live in a very cold areas are going to have cells with very specific phospholipid bilayer, comparing to us, which we have 37 degrees Celsius and we live in different environment. We're going to have different composition of the phospholipid bilayer. It is mainly the first difference between human cells and fish cells in the very cold areas is the composition of the hydrocarbon of the phospholipid bilayer. So in the previous video we were talking about that if you have more unsaturated fat inside the phospholipid bilayer, the phospholipid bilayer is going to be more resistant to cold temperature. It's going to be more fluid. Therefore, it is going to resist the cold temperature because the phospholipid bilayer won't be able to actually come closer too much. Comparing to the more viscous phospholipid bilayer, you're going to see that we have a lot of saturated hydrocarbons. Saturated hydrocarbons don't have a kink inside the hydrocarbon itself, therefore they can compact and form a lot of intermolecular forces between them. The melting and the boiling point of the following phospholipid bilayer is going to be much higher because the intermolecular forces in this phospholipid bilayer is much higher as well. Another thing that is going to compose and help us compose the phospholipid bilayer and the fluidity of the phospholipid bilayer when it comes to the plasma membrane is the length of the fatty acids, the hydrocarbons that compose the phospholipid bilayer. So the shorter the fatty acids, the less intermolecular forces you will have, therefore the more fluidity you will have inside the plasma membrane. It will be less viscous, less intermolecular forces. So it's also another very important thing to remember. One thing I would like to add is the molecule cholesterol. Cholesterol is a 4-carbon structure, 4-carbon ring structure that basically looks something like this. And it's a waxy substance that we have in many places in our body, and mainly in the phospholipid plasma membrane. Some cells the plasma membrane compose up to 50% of cholesterol inside the plasma membrane, which is very important molecule when it comes to act as the buffer for the plasma membrane. So cholesterol is a bidirectional buffer. If you have too high temperature, it is going to resist the chain fluidity and to decrease the fluidity. But if you have low temperature and it's more viscous and less fluid, you're going to also resist this state. So the more cholesterol you will have inside the phospholipid bilayer, the more resistance you will have to the actual changes in temperature and the fluidity of the actual plasma membrane. So let's sum this video up. You have 3 main things that are going to affect the fluidity when it comes to the phospholipid bilayer. One is the saturation or unsaturation of the hydrophobic regions inside the plasma membrane, which is the hydrocarbons. 2, the length of the hydrophobic regions, the fatty acids. And 3, the presence of the cholesterol molecules within the plasma membrane between the different hydrocarbons. I hope this video made sense and I will see you next time.